enzyme hexokinase uses ATP to phosphorylate glucose. This investment of energy primes glucose for energy-releasing reactions later in glycolysis. The resulting molecule is glucose 6-phosphate. ADP is released. This first step of glycolysis is irreversible. In the second step of glycolysis, the enzyme phosphoglucose isomerase catalyzes the opening of the ring form of glucose 6-phosphate to the open chain form. The same enzyme then performs a reversible reaction in which the carbonyl group of glucose 6-phosphate changes position from the first carbon to the second carbon in the chain. This reaction involves a water molecule, which donates a hydrogen ion to the carbonyl oxygen. The hydrogen ion is then retrieved from the hydroxyl group on the second carbon, creating a new water molecule. In the process, fructose 6-phosphate is formed. The same enzyme, phosphoglucose isomerase, catalyzes the formation of fructose 6-phosphate into its ring form. In the third step of glycolysis, the enzyme phosphofructokinase uses ATP to phosphorylate fructose 6-phosphate. ADP is released, and the molecule fructose 1,6-bisphosphate is formed. This third step, in which the second phosphorylation event occurs, is irreversible and is a major regulatory point in the commitment to glycolysis. Step 4 of glycolysis begins with the opening of the ring form of fructose 1,6-bisphosphate into its open chain form. In this step, the enzyme aldolase cleaves fructose 1,6-bisphosphate into two molecules. One molecule that is formed is the 3-carbon glyceraldehyde 3-phosphate. The enzyme performs additional reactions on the second 3-carbon molecule. The second molecule is dihydroxyacetone phosphate. In step 5 of glycolysis, the enzyme triose phosphate isomerase catalyzes the isomerization of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. The catalytic mechanism of this enzyme is very similar to that of phosphoglucose isomerase back in step 2. The result is two molecules of glyceraldehyde 3-phosphate. In step 6, the enzyme glyceraldehyde 3-phosphate dehydrogenase uses NAD to oxidize glyceraldehyde 3-phosphate. The resulting molecule is connected to the enzyme by a high-energy thioester bond. A molecule of inorganic phosphate displaces the high-energy thioester bond, forming a high-energy acyl anhydride bond. The resulting molecule is 1,3-bisphosphoglycerate. In the 7th step, the enzyme phosphoglycerate kinase dephosphorylates 1,3-bisphosphoglycerate. The high-energy phosphate is transferred to ADP, forming ATP. The 3-carbon molecule is now 3-phosphoglycerate. Because this reaction occurs twice, once for each 3-carbon molecule, a total of 2 ATPs are generated. At this point, the energy investment from the first 3 steps has been paid back. In the 8th step, 3-phosphoglycerate, which has a relatively low free energy of hydrolysis, is transformed by the enzyme phosphoglycerate mutase into 2-phosphoglycerate. In the 9th step, the enzyme enolase removes a water molecule from 2-phosphoglycerate, creating phosphoenol pyruvate. The loss of water redistributes energy within the molecule, creating a phosphate group with an extremely high free energy of hydrolysis. In the 10th and last step of glycolysis, the enzyme pyruvate kinase transfers the high-energy phosphate group to ADP, forming ATP and pyruvate.